Okay, next topic, Yersinia. Already Yersinia pestis is one of the organism causes plague, and other Yersiniosis. Yersiniosis means the infections caused by Yersinia pseudotuberculosis and Yersinia enterocolitica. These two are causing food poisoning and other complications also. So Yersiniosis is different. Yersinia pestis causes plague, and Yersiniosis is caused by Yersinia tuberculosis, Yersinia enterocolitica. You should know the differences. And every time, most of the students confused with Yersiniosis caused by Yersinia pestis. Here, don't confuse. Yersiniosis is different, and plague etiological agent is different. Plague is caused by Yersinia pestis, and other two species, Yersinia pseudotuberculosis and enterocolitica, causes Yersiniosis, food poisoning disease. Now coming to the Yersiniosis. Yersinia pseudotuberculosis and Yersinia enterocolitica. Yersiniosis is a zoonotic infection. Zoonotic infections means infections transmitted through the animals and pet animals, birds, all. We call it as zoonotic infections. Maybe consumption of undercooked meat and closely contact with the infected pet animals, uh, and ingestion of raw milk. So these are the source of infections from zoonotic infections. Yersinia species, that is Yersinia enterocolitica and Yersinia pseudotuberculosis. These two causes Yersiniosis. Pigs and other wild and domestic animals are the usual host. Human infections occurs due to consumption of contaminated food such as raw pork, milk, etc. It is the most common in childhood and in colder climates. Patient present with abdominal pain and sometimes with diarrhea. So geographical distribution is commonly in North Europe and America. Yersinia pseudotuberculosis generally in Finland. Serogroups, there are 6 serotypes in Yersinia pseudotuberculosis. 6 serotypes based on the somatic O and H antigens. Yersinia enterocolitica further the two groups, biotypes and serotypes. Serotypes 60 based on O antigen, biotypes 6 biotypes. And worldwide, most clinical infections are associated with serogroups 0, O is to 3 and O 9. Clinical manifestations. Overall clinical manifestations, Yersinia enterocolitica is more frequently reported clinically than Yersinia pseudotuberculosis. Self-limited gastroenteritis, diarrhea with or without blood, occurs in young children. Intestinal complications occurs in older children, characterized by terminal ileum, inflammation of the ileum. Ileitis mostly in Yersinia enterocolitica, mostly caused by Yersinia enterocolitica, and mesenteric adenitis. Patients present with acute pain abdomen may mimic pseudo appendicitis. Next, septicemia. It is typically in adults characterized with fever and leukocytosis. It usually occurs in patients with coexisting diabetes mellitus, liver diseases, and iron overload, overload of iron. Next, post-infective phenomenon occurs commonly with Yersinia enterocolitica. It occurs as a result of autoimmune activity initiated by the deposition of bacterial non-viable components in joints and other sites. Manifestations include reactive arthritis, mostly associated in persons positive for HLA-B27, human leukocyte antigen B27. Erythema nodosum, it occurs independently without any link to HLA-B27 phenotype. And Graves' disease, Yersinia enterocolitica, contains an antigen similar to thyroid stimulating hormone binding site. However, whether this cross reactivity has any significant role in Graves' disease remains unclear. Super antigens. In previous class also I discussed super antigens. Some strains of Yersinia pseudotuberculosis express a super antigen mitogen which has caused scarlet like fever in Russia and similar illness in Japan, Izumi fever, and has been linked to the pathogenesis of idiopathic acute systemic vasculitis of childhood called Kawasaki disease. Next, coming to the laboratory diagnosis. Laboratory diagnosis specimens are stool samples. Stool samples for isolation from blood culture bottles, brain heart infusion broth should be used. For isolation from lymph node aspirates, culture is done on conventional media, blood agar, nutrient agar, and McConkey agar. Blood agar, they produce granular translucent colonies with beaten copper surface, non-hemolytic colonies seen. McConkey agar, growth of Yersinia pseudotuberculosis is poor. Yersinia enterocolitica grows well and produces lactose non-fermenting pale colonies. For isolation from feces, food, or soil, selective media should be used such as deoxycholate citrate agar, McConkey agar. McConkey agar, it shows non-lactose fermenting colonies, and another selective media, Yersinia sin agar, cefsulodin irgasin novabiocin. Sin means cefsulodin irgasin novabiocin agar. Colonies on sin agar, cefsulodin irgasin novabiocin agar, it shows colonies are looking like bull eye colonies. Bull eye colonies. Incubation, plates should be incubated at 25 degrees Celsius and 37 degrees Celsius to differentiate from the most of the other pathogens which grow only at 37 degrees Celsius. Cold enrichment can also be done by incubating in phosphate buffered saline at 4 degrees Celsius for 3 weeks. Next, Yersinia enterocolitica and Yersinia pseudotuberculosis shows the following properties by which they can be differentiated from pestis. Differential motility. They are motile at 22 degrees Celsius but not at 37 degrees Celsius. Yersinia enterocolitica and pseudotuberculosis shows they are motile at 22 degrees Celsius, but Yersinia pestis 37 degrees Celsius, it shows the motility. Next, cold enrichment. Growth improves on refrigeration 4 degrees Celsius. Automated identification systems such as MALDI TOF can also be used for accurate species identification. Serology, antibodies can be detected by agglutination or ELISA using serotype specific O antigen type. In pseudotuberculosis infection, antibodies appears early during acute phase of illness, whereas Yersinia enterocolitica species agglutinating antibodies are more likely to be found in convalescent serum. Next, treatment. So, Treatment of this Yersiniosis, most cases of diarrhea are self-limiting. Treatment is required only for systemic infections such as in case of septicemia. Fluoroquinolone, ciprofloxacin or third generation cephalosporin, cefotaxime are mostly effective. For Yersinia enterocolitica strains nearly always produce beta lactamase but not Yersinia pseudotuberculosis strain. Okay. Okay. Thank you.